Hello everyone, welcome to IAS Baba 60 days rapid revision series for Prelims 2022. This is day 17 and today we are taking up science and tech. So these are the topics we are going to discuss, and before that we have some doubts to be cleared. So first one was the elasticity. So elasticity is not the ability of an object to deform, but once it deforms, the ability of the object to come back. So that is the proper definition of elasticity. So that means, so if an object is not brittle, so if it is not brittle, the solid is having the highest elasticity. So if it brittles, it breaks. So else it comes back to the original form. Then come to next, the Committee on Public Sector Undertakings. So had given a old data. So we will take it as 22. So it is like 15 from Lok Sabha and 7 from Rajya Sabha. So 15. So 15 is the number when it was started. Okay, in 1964 when it was started. So that was the number. So anyway, we got one additional information here. Then Shah Jahan 3. So when the third battle of Panipat was going on. So it was like the battle was going on between the Shah Jahan III and Shah Alam. So there in that context, so he was being overthrown at that point of time. So he was not ruling at that point. So he was overthrown. Then come to next, the suspension of members. So here also, like, yes, it was true that as per the rule 255, it is like once suspended. So it is only for that day's meeting. So the next day, the MP can attend the meeting, and then if a MP is suspended, so he can be that suspension can be terminated with another motion. Okay, so both are correct as you have suggested. Then come to next, the first one, CRISPR-Cas9. Friends, CRISPR-Cas9 is also called as molecular scissor. So in genetic engineering, so this cuts the molecules. So here, what is this CRISPR-Cas9? CRISPR means clustered, regularly interspaced palindromic repeats. So that means it is cutting exactly at the palindromic repeats. So what is a palindrome? Palindrome is a word wherein we read both either from right or left. So it pronounces the same. Say for example, madam. So it is a palindrome because you read either ways. So we get the same pronunciation. So the same way, if we have two genes, so gene 1 and gene 2. So in this double-stranded DNA, so in the middle, so here we have a palindrome. Say for example, we can have ATTA. So this is a palindrome. So CRISPR-Cas9. So Cas9 is the protein. So this exactly cuts the palindrome here. So that is why it is called the CRISPR, that is clustered, regularly interspaced. So these palindromes are regularly interspaced because they appear after every gene. So clustered, regularly interspaced palindromic repeats. Okay, so don't by heart. So understand the things. Then come to next, 3-parent babies. So here the mitochondrial DNA disorders. So these are the unique disorders mainly present in UK. So that is why UK government had given permission for these 3-parent babies. So here the mitochondria, so they will be defective. So we take one egg which is having a disordered mitochondria, and out of that egg, so we will transfer the nucleus and we will put it into a healthy egg. So a healthy zygote will be inserted with this nucleus, and the nucleus of this healthy zygote will be discarded, and then this will be developed so into a baby. So now this baby will not be having any mitochondrial disorders. So here in 2021 prelims, so two statements had come. One is that, can we do this nuclear transfer? So even before and after fertilization? Yes, we can do it before or after fertilization. Say for example, I have taken one zygote, so that consists of both mother and father's gene, and I will transfer the nucleus. So I will take one more zygote, so which is the donor one. I will discard this nucleus and I will add this here. So now I will get the new zygote, so which is having a healthy mitochondrial DNA. So before fertilization, can I do it? Yes, I can do it. Now I will consider a mother's egg. So here this egg, it is having a disordered mitochondria. So now I will take the nucleus out of it and I will take a donor egg. So I will discard this donor egg and then I will introduce this one into here, and now I will get an egg. So here this egg will be having the healthy mitochondria. Now I will fertilize. Okay, so when I fertilize this egg now, I will get a zygote. So this will be having the healthy mitochondria. So here it was before fertilization and here after fertilization. So likewise, both the things can be possible. Okay, so one more statement was there, that is the mitochondrial DNAs, so that are derived entirely from the mother cell only. Yes, it is derived from entirely from the mother cell only because, so the mitochondrial DNA, it lies only in the protoplasm. So protoplasm or within the cytoplasm it lies. So these mitochondrial DNAs which are present inside the cytoplasm, they have nothing to do with the nucleus. So they don't have any connection, but the father, so he will give some content only to the genes, and that will be restricted to within the nucleus. Only within the nucleus we have father as well as mother. So outside the nucleus, it is all about the mother who is contributing. Okay, so both the statements are correct. Give importance to the concepts first and then give importance to the extreme words. So just because they are extreme, don't hit blindly. Then come to next, induced pluripotency. So before induced pluripotency, we take up pluripotency first. So here like we have the stem cells. So stem cells are the ones which can differentiate and replicate into various other kinds of cells. So here I have a zygote. So fertilization is going on. So this zygote, so that will form two kinds of cells, that is the blastocysts and the baby cells. So this cell has the ability to differentiate into blastocyst cells and the baby cells. So that is what is called totipotent. And then we take up these baby cells. So blastocyst, so that will become the amniotic sac. So inside which the baby will be covered. So that will nourish the baby. And now we take up the baby cells. So these baby cells, they can be differentiated into ectoderm, mesoderm and endoderm. That is the inner tissue, middle tissue and outer tissue of a body. So that is why these baby cells are called pluripotent cells. And then here in the ectoderm, we can see this ectoderm can form either nerve cell or the skin cell, and this mesoderm can form either a blood cell or the muscle cell. So that means they also have some ability to specialize. So they are called multipotent cells. And then lastly we have these nerve cells, red blood cells, then liver cells, pancreas. So all these, they don't have any ability to specialize. So they have already specialized and they can replicate only their own kinds of cells. A red blood cells can replicate to another red blood cells only. So they are unipotent cells. So this is the hierarchy. So remember, totipotent, pluripotent, multipotent and unipotent. And now how to induce? Say for example, I have taken a unipotent cell, and how will you make it as a totipotent? How will you make it as a pluripotent? So that has to be done artificially with the help of genetic engineering. So earlier it was not being possible, but later Shinya Yamanaka, a scientist, so he came up with 4 genes. So these are the 4 genes, that is OCT4, SOX2, CMYC and KLF4. So he said that if I introduced all these to a unipotent cell or any specialized cell, so I will get a stem cell. Say for example, he introduced these factors, these genes into a skin cell, and then he got a pluripotent cell. So that is able to get differentiated into any kind of cell like this. So that artificial inducement of pluripotency is called induced pluripotency. And remember, these are called Yamanaka factors or the reprogramming factors. And then come to next, the Saline gargle RT-PCR technique. So here it is like earlier, whenever we go for the RT-PCR, so the sisters and the doctors, so they used to take the swabs from our nasal tract or they were taking swabs from our sputum. So that was a difficult task. So that required some medical experts. But later, Government of India, CSIR and NEERI, so they came up with this saline gargle RT-PCR. So saline gargle means that they will give you a solution, you will gargle that and you will spit it into a test tube. So that itself becomes the swab now. So here we need not require the front-end doctors. Friends, we know that front-end doctors are the worst victims of COVID-19. So that's why to avoid that, so this saline gargle RT-PCR technique was introduced. So this is the difference between normal RT-PCR and the saline gargle RT-PCR. Now a brief discussion on RT-PCR. So what is RT-PCR? It is reverse transcription polymerase chain reaction. So reverse transcription means so conversion of RNA to DNA. Polymerase chain reaction means multiple replications of this DNA. So now we take the swab and in that swab there will be COVID-19 RNAs. So if there are COVID-19 RNAs in that, so then we will be having one synthetic RNA. So that will go and bind to this COVID-19. So if there is no binding that is happening, so that means that there is no COVID-19 in that swab. Okay. So if it binds, so then here in this we will be having a fluorescent tag, and now we will replicate this repeatedly. So when we replicate this repeatedly, so here we will be having so many fluorescent tags, and now this liquid starts glowing with fluorescent color. So because there are so many tags, because it has been bound there, so now more fluorescent color means it is COVID positive. So this is how the RT-PCR used to work. And then the Saline gargle RT-PCR, it has several advantages, and remember it was being prepared by CSIR and NEERI, that is National Environmental Engineering Research Institute. So here some of the advantages like the skilled healthcare workers are not required for collection of samples, and then the collection of sample, the process is non-invasive, and then the waste generation can be avoided. So that is the extra sputum, extra mucus, so all those things will be avoided here. Then come to next, the Feluda test. Friends, when RT-PCR was going on, so RT-PCR was time consuming. So at that point of time, the Feluda test was introduced. So this was indigenously produced by Tata-based research institute. So here Feluda is a strip test. So that detects the coronavirus in 1 hour. So within 1 hour you will get the result. So here in the RT-PCR, that polymerase chain reaction, that is time consuming. So that multiple replication is time consuming. So if we cut that, so then we will get the test report early. So here we got this technique. So that Feluda is an acronym for FN-Cas9 editor linked uniform detection. So Feluda, so that is how. So Feluda is also the name of detectives is called Feluda in Bengali. So we can add that also. So here how this Feluda test actually happens. So first the RT-PCR goes, but the polymerase chain reaction doesn't happen so much here. Say for example, here we have a COVID RNA, and then the reverse transcription happens. So the DNA is formed, and now to some extent, so the polymerase chain reaction happens. So we will say like 4 to 5 replicas of DNA are obtained. And now the Cas9, it is being allowed into this. So we add Cas9 protein into this. So now what Cas9 does? So Cas9's duty, CRISPR Cas9's duty is to cut the DNA. So now it starts cutting DNA into various fragments. So friends, whenever the DNA is being cut, so whenever gene addition takes place, so much of ions are released. So these ions, so they will indicate the presence of COVID-19. So now how you will detect that ions? So that ions will be detected with the help of a pregnancy strip. So pregnancy strip, so they will detect the ions because pregnancy strips, they will be having gold nanoparticles in front of them. So whenever ions are created, these gold nanoparticles, they will vibrate. So now with the help of pregnancy strips, we are testing this COVID-19, and that is why it is the Feluda test. So remember, it is indigenous one and it is related to genetic engineering. So that's why it becomes important. So here we have the pregnancy strips. So with the help of these strips, we are going on with the Feluda test. Then come to next, world's first genetically modified rubber. So here the GM rubber, so that was developed by the Rubber Research Institute of Kerala. So that is Kottayam, and then it was planted at the Rubber Board's Sirutari Research Farm in Guwahati. So it was prepared in Kottayam, but its practice, manifestation or the growth, so that happened in Assam. So here what was the gene? So the gene was the MnSOD or the manganese containing superoxide dismutase. So what is the function of this gene? It is that cold tolerance. So once we introduce this gene, the rubber plant will be tolerant for cold. Friends, we know that the rubber is a tropical plant, so it doesn't tolerate extreme colds. So now if we add this gene, so now it becomes tolerance for the cold. From where that gene was taken? It was taken from Pisum sativum or the pea plant. So common pea. So we know the peanut butter. So from that pea plant, this gene is being taken. So remember this. And then the pig heart transplant in the human patient. So recently we know that the pig, the genetically modified pig was there. From that, the heart was taken up and that heart was introduced into a person. Okay. And that person, okay, so he died after 2 months. Okay. So it was like this pig was genetically modified so that if I take an organ, say I take kidney, heart, etc., and then if I introduce into the humans, the autoimmunity will not be triggered by the human beings. So earlier, if I take any heart or a kidney from natural pig, so that will trigger the immunity and we have to suppress that immunity. So now this GMO pig, so it is a specific kind of pig where its parts will not trigger autoimmunity. So that is a great advantage, friends, because the transplanted people, they will suffer a lot if we give the anti-immunity or the anti-immunity medicine or the immunity suppressants. So they have high side effects. So to avoid that, we tried one experiment. So but it was not so successful. Okay. So this is also called as Xenotransplantation. That means transplanting an organ from animal to human beings. So here the special thing was, it was a GM pig. Then come to next, autosomal DNA technique. So we know that the sex DNA and autosomal DNA. So we have 46 chromosomes in the human beings, and in that 23 pairs. Okay, so in that 23 pairs, 2 are sex chromosomes, that is X and Y or X and X. So the remaining all chromosomes, so they carry autosomal chromosomes. So that means apart from sex, all other characteristics, so characteristic of a hair color, body height, so all these, they belong to autosomal DNAs. So here, this autosomal DNA technique, so that is used to find the heir or to find the descendant of Mr. Lakota. So Lakota, he was the person, so he was the leader of the tribe which ruled the United States before the Europeans invaded US. So before they invaded, so he was the person who ruled them. So his descendant, so he was being found out by this. So friends, mainly the pedigree analysis. Pedigree analysis is a system where we will analyze our ancestors, fathers, great-grandfathers and others with the help of DNA analysis. So here this autosomal DNA technique was used. So why this autosomal DNA technique? Because whenever we don't have any sex chromosome to detect the DNA, so but obvious we will have this autosomal DNAs. So these autosomal DNAs, they are not the actual detectors of the ancestors. So here we will be getting only few genes which correlate to our father or mother. And now the technique goes like, so if you have some genes, so that will be taken up, and if someone else has some specific genes, so they will be taken up, and now these two will be compared. And now here, if there are any differences, they will be removed and one set of gene will be remaining, which are absolutely common. So likewise, the mixture and taking and extracting of DNA, so those things have happened. So we say that instead of going for the gene chromosomes, so autosomal DNA, autosomal chromosomes are used for this technique. So that is how the pedigree analysis has been done in this process. So remember all these and remember autosomal DNA technique. And then experimental advanced superconductor Tokamak, that is the Tokamak. So here we know that this Tokamak is nothing but artificial sun. Okay, so we have China going up with the artificial sun. Artificial sun means the nuclear fusion reaction. So that is manifested here. Friends, we know that in the nuclear fusion reaction, so we require very high temperature, and if we require high temperature, so that chamber in which we are performing that reaction, so that should be having a very strong magnetic field. Okay, so for strong magnetic field, we have constructed one Tokamak. So Tokamak is a chamber within which the nuclear fusion reaction takes place. So that will be in the form of duff nut. So that means it will be having a rounded shape with the hollow in the middle. So here in this shape, the reaction occurs. Okay, so we had built a chamber in China and we had tried for this, the nuclear fusion reaction. So if we try the nuclear fusion, so that means, so we are coming up with artificial sun. Okay, so this East project, so that is a part of the International Thermonuclear Experimental Reactor. So now if all these are succeeded, so then but obvious we are going to have one artificial sun or might be a successful sustainable fusion reaction. Okay, so we will update in future if all these succeeds. Okay, so Russia, China, then South Korea, then Japan, United States. So all these are doing their own experiments in this fusion reaction, and in future we will update. Then come to next, gamma ray burst detected by NASA's Fermi Gamma Ray Space Telescope. So here the NASA's Space Telescope, so that will work in this gamma ray. So they have detected some gamma ray bursts. So bursting of the gamma rays. So they are also called as GRBs or the gamma ray bursts. So they are immensely energetic explosions, so that have been observed in the distant galaxies. Okay, so from the galaxies these rays are being coming. Then they are the brightest and the most energetic electromagnetic events known to occur in the universe. So that most energetic and the brightest, so mark this as important, they are not extremities, they are factually correct. Then bursts can last from 10 milliseconds to several hours. Then after an initial flash of gamma rays, a long-lived afterglow is usually emitted at a longer wavelength. So but obvious, so here after one glow, so that radiation goes on with its radiance. Okay, then the intense radiation of most observed GRBs is thought out to be released during the supernova. So supernova is a process wherein when a star dies, so it will explode. Okay, so at that stage, these gamma ray busters or gamma ray bursts, so these are going to be emitted. Okay, so here all observed GRBs, they have originated from outside the Milky Way galaxy. So all those soft gamma rays, so they are being associated with Milky Ways. So here the overall meaning of these gamma ray busters are that, so whenever a star undergoes supernova, so some gamma rays, intense the energetic radiations are being emitted, and now we have tracked these from a galaxy that is outside Milky Way. Okay, so we have an hypothesis that these gamma rays, so they are going to destroy the Earth, so they are going to destroy the whole universe. So this is the hypothesis because they are so energetic with that. Okay, so now we should be careful that if any stars nearby us, nearby the sun, if they undergo supernova, then we will make sure that how we will save the Earth from these gamma ray busters. Okay, so the gamma ray bursts and the energetic and the destruction of the Earth. So that, if you remember, that is more than sufficient. Okay, so then coming to next, India's 3-stage nuclear program. So why it was in news? Because India's prototype fast breeder reactor, so that went critical recently. Okay, so in that perspective, we will make sure that this will be studied. Okay, so now make sure that which was that prototype, where it was present. So you can comment that in the comment box. So which was the fast breeder reactor? Friends, we have the pressurized heavy water reactors. So that was the first stage of Indian 3-stage nuclear program. Slowly we will move towards the fast breed reactors. So if we move towards fast breeder reactors, so then we need not purchase the nuclear materials from other nations. So then we will be having the thorium based nuclear reactors, and we have excess of thorium in the Kerala's monazite sands. So we will be self-reliant in the nuclear technology. So here we go with the stage. So again we have a brief discussion. We need not go into that detail. So UPSC doesn't ask our specifications, only the conceptual clarity. So at the stage 1, so here we are using uranium-235, and along with uranium-235, uranium-238 also goes. And this uranium-238, so this will give rise to plutonium-239, and this plutonium-239, so that will be fed to the second stage, and along with that, the thorium is also fed. Okay, so we are making this reactor habituated for the thorium. So that is why we are going for a smooth transition. So suddenly if you add thorium, so the criticality will not be obtained. Okay, so here from uranium, then plutonium, and thorium is added slowly. And then coming to next, so the uranium-233, so that is obtained here, and that uranium-233 is fed, and then thorium is fed. So this uranium-233, so it is very minute in the third stage, and maximum fuel comes from the thorium. So now this reactor, okay, so this reactor is the one which sustains from thorium. So we require the same. Okay, Homi Jahangir Baba, he came up with this idea, and we are already on the journey to come up with this purely thorium based reactors. Okay, so this is all about the 3-stage reactors. And then the IAEA and JCPOA. So here we first see the context. Iran has informed the International Atomic Energy Agency that it intends to produce uranium enriched up to 20% purity, well beyond the threshold set by 2015 Vienna Accord. Okay, so Iran has rebelled now. It says that I am going to enrich more, so do whatever you want. Okay, so it is the JCPOA, so that is officially called as the Joint Comprehensive Plan of Action, and it was signed between Iran and P5 plus Germany. So P5, the permanent members of UNSC, and then Iran agreed to cut its stockpile of low enriched uranium by 98% and reduce by about 2/3 the number of its gas centrifuges for 13 years. So centrifuges are the ones which are used to enrich uranium. Okay, then for the next 15 years, Iran will only enrich uranium up to 3.67%, and Iran also agreed not to build any new heavy water facilities for the same period of time. Okay, so these were the promises made by Iran in this treaty. And then come to next, a brief discussion on IAEA, that is International Atomic Energy Agency. So firstly it was called as Atoms for Peace. Okay, so that was established in 1957, and it has its headquarters in Vienna, okay, Austria. Then the primary mandate of organization was and continues to be promoting safe, secure and peaceful nuclear technologies. So this was the aim, and currently it has 171 members, and the latest member is Saint Lucia, so which joined IAEA in 2019. So mark this. And then India became a member in 1957 itself. So India was a member at its inception. So many times we get this statement in UPSC. So India was a member at its inception. Yes, it was. So mark this as important. And then nuclear suppliers group. So that was brought in 1974 in response to India's nuclear test, Smiling Buddha. Okay. So we tested this. So that's why it was. And then it is a multilateral export control regime. Okay. So it is like here, if we are in a nuclear suppliers group, so there is a restricted transport of nuclear element from amongst the members to the outsiders. Okay. So it is a group of nuclear supplier countries that seek to prevent nuclear proliferation. So it is a group of nuclear supplier countries, and the NSG first met in November 1975 in London and is thus popularly known as the London Club. So London Club is another name of the nuclear suppliers group, and it is not a formal organization and its guidelines are not binding. So mark this, not binding. Then decisions including on the membership are made by consensus. So decisions are made by consensus. So it is basically a group of nuclear suppliers, and whenever these nuclear suppliers, when they supply nuclear to the non-members, so they will be taking some precautions. Okay. Then come to next, the NPT, the Non-Proliferation Treaty. So it is a multilateral treaty signed in 1968 and entered into force in 1970, and it has 190 members. Then the treaty defines nuclear weapon states as those that had manufactured and detonated a nuclear explosive prior to the 1st January 1967. So these, they have the ability to have and own the nuclear weapons, and others they cannot, and all the other states are therefore considered as the non-nuclear weapon states. And then the provisions, the 5 nuclear weapon states are the China, France, Russia and United Kingdom and United States. And then the treaty does not affect the right of the state parties to develop, produce and use nuclear energy for the peaceful purposes. So these states, so they can have the nuclear arms, and others they cannot have the nuclear arms, but they can have nuclear only for the peaceful purposes. That is, they can have electricity out of the nuclear reactors, but they cannot build any nuclear weapons. So this was partiality. That is why only these 5 nations should have nuclear armaments, why others cannot. So that is why India was also against it, and India, North Korea, so we didn't sign that treaty. Okay. Then nuclear weapon states are not to transfer nuclear weapons into any non-nuclear weapon states. So the nuclear weapon states should not transfer any weapons to the non-nuclear weapon states, and the non-nuclear weapon states must accept the International Atomic Energy Agency safeguards on all nuclear materials on their territories or under their control. So if we are having any nuclear material, so that has to be certified by IAEA. Okay. So these are some of the clauses of NPT. Then come to next, CTBT, that is Comprehensive Test Ban Treaty. So that entered into force in 1996, and the CTBT aims to prevent further horrendous health and environmental damage caused by the nuclear test explosions. So here it is like by testing the nuclear weapons, so much of damage is being caused. So that is why CTBT, it concerns only with the testing, and then it curbs the development of new nuclear weapons and the improvement of existing nuclear weapon designs. Okay. So it is like both the armamentation and the testing, because if you are coming up with any new nuclear weapon, so but obvious you are testing that. So again, both are interlinked. Okay. Then come to next, the New START doctrine. It is a nuclear arms reduction treaty between the United States and Russian Federation. It entered into force on 5th February 2011, and it replaced the Treaty of Moscow, or the SALT, which was to expire in December 2012. And its name is a follow-up to START 1 treaty. So we know that when the START 1 treaty happened, START 2 and START 3, so they were also conceptualized, but START 2 and START 3, so they didn't materialize. And then under the terms of the treaty, the number of strategic nuclear missile launchers will be reduced by half. Then a new inspection and verification regime will be established, replacing the SALT mechanism. So SALT was earlier, now we are going to have a new mechanism between US and Russia. Then the number of deployed strategic nuclear warheads is limited to 1,550. Then the number of deployed and non-deployed intercontinental ballistic missiles, submarine launched ballistic missile launchers and heavy bombers would be limited to 800. So including all these ICBM, SLBM and heavy bombers, so totally not more than 800 should be held. And then come to next, the timeline to meet these targets. That is, these obligation must be met within the 7 years from the date the treaty enters. So 7 years is the timeline. Then the treaty will last 10 years with an option to renew it for up to 5 years upon the agreement. Okay, so like up to 10 years it stays, and if want, we can extend it to 15 years. That means 5 more years. Okay, so these are some of the facts, and remember the concepts and try answering more and more mock questions on those concepts. Okay, so that you will get various angles and you will have a command on the concepts, and make sure that you will revise the facts so that you will not forget them. Okay, so these are some of the things. Coming to the last part, friends, again, it is like whenever you come up with a difficult concept, so it requires some necessary hard work to understand. So it is not like we sit idle and the concept will come on its own. So that ability to have a command, it requires necessary hard work. So that sweat and blood if you have, so nothing is impossible in this world. So make sure that if you have that courage, dedication and determination, okay, so everything you are going to achieve, so that box down to you and that salutes to you. Else you will have to salute not only for that, you will have to salute for everyone. So make sure that you have that dedication. Do it all the very best. Good luck, friends.